A very good morning, ladies and gentlemen. Brief about the Department of Biotechnology Engineering, NMMIT Nitte. Bachelor of Biotechnology Engineering program started in 2002 with an intake capacity of 60 to train students in biological applications of engineering principles. We are autonomous attained under VTU Belagavi from 2007 onwards. We also have been accredited by NBA between 2012 to 2017 for 5 years. Highlights of the department includes VGST Center of Excellence. We are involved in a number of interdisciplinary projects. We have student exchange program with foreign universities. We are continuously involved with students and faculty research programs wherein we have a total grant received amount to 2.5 crores with more than 150 publications in reputed journal. We have experienced faculties and all the faculties have completed their PhD programs. Our laboratories are well equipped with various equipments. Students and faculty are continuously involved in competing with day-to-day challenges in terms of research and development, have received awards from different authorities. NMAMIT Biotechnology Department is one among the 32 information and demonstration centers instituted by Karnataka State Bioenergy Development Board with an annual grant of rupees 4 lakhs per year. The center, along with the production of biodiesel from various non-edible oilseeds and used cooking oil, is available in our campus. It also creates awareness, disseminates information about biofuels amidst scientific community, students, farmers, and public, and also demonstrates the technical know-how of biodiesel production. This center was established in the year 2011. Ladies and gentlemen, our conference, Nitte-Bio, Nurturing Innovative Technological Trends in Engineering Bioscience. This conference is expected to display a significant development in the important areas of biotechnology and their relevance to the welfare of mankind services. Conferences thus provide a valuable opportunity for research scientists, industry specialists, and decision makers to share their experiences. It's my pleasure to welcome all the participants, paper presenters, authors, faculty researchers, students, and industry experts from across the globe for this international conference, Nitte-Bio 2021. I take pleasure in extending a warm welcome to the eminent speakers around the world. Ladies and gentlemen, we have Mr. Ibrahim, Vice President Novo Nordisk from Denmark, as a keynote speaker. Dr. Jairo Quijano, Professor from University of Columbia, Medellin. We have Ms. Alina Semkina, the Chief Commercial Officer from Russia, and Dr. Sriram Shetty, Professor from Texas, USA. All these experts will be sharing their research expertise, their views in their fields of excellence with us. I also would like to welcome all our session chairs who will judge the presentation sessions. An exceptional diversity of the applications of biotechnology will be revealed by the researchers in these two-day event, which would run in parallel sessions, wherein original research paper will be presented by researchers all over the country. A total of 48 papers were accepted out of hundreds received, which will be presented in this 2 days conference. Various streams of this conference do include enzyme technology, bioprocess design and engineering, green synthesis of nanoparticles and their applications, pharmaceutical biotechnology, cancer biology, bioremediation and biofuels, and bioinformatics. I hope This conference will enhance your knowledge on novel concepts and thereby stimulate your mind to dwell into further research. I wish all the participants every success with this conference, and I look forward to learning about the outcome. I once again extend a warm welcome to all of you. Thank you and all good wishes from the biotech family of NMIT Nitte. Thank you, one and all. Professor Jairo?
Hi.
Hi, Professor Jairo. Am I audible? Professor Jairo?
Okay, nice to meet you.
Yeah. A very early good morning to you from your Colombia and a
Hi.
late good morning from my side
Professor
from
Jairo.
India.
Am I
We are
audible?
very
Professor
delighted
Jairo?
to have you,
Okay, nice to meet you.
and I'm very thankful to
Yeah.
you for
A very early good morning to
uh,
you
your
from your Colombia
presentation,
and
which you
a
would
late
be
good
further
morning from my
presenting
side from
in front
India.
of all
Am I
the
audible?
very
Professor
respected
Jairo?
participants
Okay, nice to
With
meet you.
and
our
our
very
international
thankful to
conference
you
being held for
for a
2 days.
very early good morning to you.
And now
Your
I would
Colombia and
just
a late
be
good
further
morning from my
presenting
side from
in front
India.
of pass on the
Am I
system to
audible? Professor
the
Jairo?
participants.
Okay, nice to meet
Finally,
you.
Our
our
very
international
yeah,
conference being held for
a
2 days.
very
For a
early good
brief
morning to you.
introduction
Your
now, I would
Colombia and
just
a late good
eminent
morning from my
speaker
side from
from
India.
Columbia Medical
Am I
System
audible?
to
Yes, Professor
the
Jairo?
participants.
Okay, nice to meet
Finally,
you.
our
Our very
international conference being held for
a
2 days.
very
For a
early good
brief
morning to you.
introduction
It is
now,
my
I
Your
would
immense
Colombia
honor
and
just
a
to
late good
eminent
morning from
introduce
my
speaker
side
Dr.
from
from
India.
Tihanu.
Columbia Medical
Am I
Dr.
System
audible?
to
Yes,
Hayati
Professor
the
Tihanu
Jairo?
is working as a
participants.
professor
Finally,
in
our
Columbian
registered
Polytechnic,
bioinformatics
JIT,
Very
for a
Medellin,
good
brief
morning
Colombia.
and
He
now
completed
I
his
would
engineering and
just
MSc in
eminent
physics from
speaker
National
from
University of
Columbia
Colombia
Medical
Am I
System
in
audible?
to
Professor
the
2004
Jairo?
and
participants.
2007
Finally,
respectively in
our
Columbian
registered
Polytechnic
A
from
bioinformatics
the
very
Swiss
early
Federal
good
Institute
morning to
of
you.
Technology
He completed his
in
engineering
Lausanne,
and MSc in
Switzerland
physics
in the
from
year
National
2003
University
of
of
Colombia.
Am I
In
audible? Professor
2004,
Jairo?
20 research articles
Finally,
in
our
various
registered
peer-reviewed journals.
very
His
early good
area of
morning to you.
expertise include
Now I would
cell and
just
cancer biology,
from
BIOscience,
National
and plant
University of
protection.
Colombia.
In
Am I audible?
2004,
Professor
20
Jairo?
research articles
Finally,
in
our
various
registered
peer-reviewed journals.
very
His
early good
area of
morning to you.
expertise include
Now I would
cell and
just
cancer biology,
from
BIOscience,
National
and plant
University of
protection.
Colombia.
In
Am I audible?
2004, 20 research articles
Finally,
in
our
various
registered
peer-reviewed
Okay.
journals.
very
His
early good
area of
morning to you.
expertise include
Great to
Now
be
I would
here.
cell and
I'm
cancer
gonna
biology,
from
BIOscience,
National
and
present
plant
my
University of
protection.
Colombia.
In
Am I audible?
2004, 20 research articles
PowerPoint present.
Finally,
in
our
various
registered
peer-reviewed
Okay.
journals. His
very
Okay, so
area of
early
please
good morning
expertise
to you.
confirm
include
to
Now
be
I would
here
cell
and
and
see.
cancer
Gonna
biology,
from
BIOscience,
National
present
and plant
my
University of
protection.
Colombia.
In
Am I audible?
2004, 20 research articles
PowerPoint
Visible?
present.
Finally,
We
our
we can see the
registered
presentation.
peer-reviewed
Okay.
journals. His
early
So
good
area of
morning
please.
to you.
expertise
Okay,
include
Now I would
here.
cell
Here
and
we go.
cancer biology,
So first of all,
BIOscience,
present
and plant
my.
protection.
I'm gonna thank the
Yes,
International
visible.
Conference on
We can see the
Nurturing
presentation.
Innovative Technologies. Okay, technological trends in engineering for the invitation and big thanks to you. I'm gonna thank our usual
Yes,
International
visible.
Conference on
We can see
Nurturing
the
dating this
presentation.
innovative technologies. Okay, technological trends in engineering for the invitation and big thanks to you. So usual interrogating this of the camera technology. Okay, logical
Yes,
principle
Professor.
in engineering for representation and visualization. So to focus on the presentation, interrogating this of the camera technology. Okay,
Yes,
okay.
Professor.
So this presentation is entitled UVC radiation for controlling gray mold disease in study process. The first
Yes,
thing to
Professor.
say is that this project presentation is entitled UVC radiation with tropical plant breeding research group here in Medellin, Colombia. The first thing to say is that this project I am the director radiation with applied physics breeding group. So this work here in Medellin, Colombia, done in with these two groups in collaboration. Here we will have the opportunity to see mainly how different sciences are combined with engineering to bring like alternative solutions. We will have the opportunity to see mainly agriculture issues. How in this case science are controlling engineering to bring fungi alternative solutions, basically known as agriculture how in this case science are controlling engineering to bring fungi alternative solutions, basically known as agriculture how in this case. So we're gonna have an introduction, a brief introduction into the subject that we touch here in this presentation. And the first thing is just to realize that the fungi are the greatest threat we touch here in this presentation. And the first thing right here is just to realize that tomatoes in the greenhouse are the greatest threat like touch here in messed up with and the first thing fungi and traditional tomatoes kind of greenhouse agricultural methods have been treated with synthetic chemical fungicides. Normally, that is the traditional greenhouse. So we all know, or we can guess, that these have been treated with chemical approaches. Are really normally invasive. That is the traditional plant itself, but also the environment. And if you are not really careful, finally the people that operates these crops are affected too. So this is one main concern. The other concern is that this like
Professor, I'm sorry to disturb you, Professor, but
yeah,
we are not able to see the presentation if you have already started it. We are not
oh,
able to see the slides, please.
okay, wait. Mm, right now you can see it, or
Yes, Professor, yes.
yes,
Perfect, perfect.
okay, okay. So again, you can see this image of a tomato greenhouse plantation. Okay, so I was saying, we know that fungi is the greatest threat to food production. All right, this is treated normally with synthetic chemical fungicides that affects the environment, the human health, also the soil, and finally, we know that these fungi have the ability to develop a resistance to these chemicals. So this implies that if you're using chemicals, afterwards you need to like increase this the concentration of these chemicals, and this unfortunately is um, is like a cause for a public health issue and high risk for the environment. this has forced the industry to search and develop alternative methods for disease management. This is mainly the cause for searching alternative methods. So one of these alternative methods is like a physical approaches. So there have been also other approaches like biological methods, and here we're gonna focus just in one physical method, which is the use of light. As simple as that. It's just using light. In our case, is using UVC light. So since we're gonna talk about a little bit of light, I can just explain some basic things about light. So light is an electromagnetic wave, and electromagnetic wave has an energy which is given by this formula, which is h, the Planck's constant, and nu, which is the frequency, or in other version, frequency and wavelength are inverse. So basically, uh, light is energy, right? It's a wave that carry out energy and is quantified by the wavelength. And here we're gonna have just light interacting with matter. In this case, biological, meaning the fungi or the plant. So there is a propagation of photons that are the carriers of the light. And okay, think more about that. The important thing is to understand that the wide spectrum of electromagnetic wave, which is called light, which is not called light, but um, overall, an electromagnetic spectrum. Here we have it as a figure that can help us understand somehow the relationship between the wavelength, the frequency, and some images that we can be familiar to. So here we're gonna be in this region, the region of the wavelength of of more or less the size of molecules, and the frequency implies that the energy is really high, not as high as the X-rays or gamma rays, but it's pretty high. And we can relate this to also compare it with another electromagnetic waves that are used in medicine, for example, like X-rays or gamma rays, or even infrared are used too. So here we're gonna focus on this, just a small part of the electromagnetic spectrum that is called ultraviolet. And even in the ultraviolet spectrum, we're gonna focus just in the C part. So before that, we need to understand that a light or any electromagnetic wave can interact with matter in different manners. So basically two. There is light that can cause ionization of molecules, and light that do not cause this. So here the ultraviolet, the UV light is just in the limit in between the energies that can cause photoionization like X-rays and gamma rays, and the ones that do not do that, like the visible light or infrared or microwave, that is a common electromagnetic wave used for heating our food. Okay, so this means that this kind of wave, ultraviolet UV light, is is in the limit of being able and not being able of cause some ionization. So this is a very, very important aspect of this light. Okay. Now focusing the UV, we can split this UV in three parts. UVC, which is coming from 100 to 280 nanometers. UVB, which is 200 between 200 and 315 nanometers, and UVA, which is the UV light, which is closer to what we call visible light, which is where are able to see as or we consider visible light. Okay, so here this picture shows that the UV light is mainly the source of the UV light is mainly the sun. So this is an important thing to have in mind because the ozone layer can let pass just a few UVA and UVB, but UVC, the ozone can absorb it. So mainly here on Earth, we cannot have UVC light coming from the sun. This is also important to notice the high concerning about the ozone layer, right? Because the ozone layer is making this work of not letting this highly energetic light to come to us. So, all right. So that means that if we want to have an UVC light source, we need to produce it. So how to produce UV? How does it work mainly? So we have sources of UVC light, like artificial ones, coming from just lamps, two different kind of lamps. Maybe the one on the center is produced by LEDs, and the one on the left is produced by a gas, so a lamp gas, or what is called a germicidal lamp. Okay, how does it work? So basically, it's kind of like the wavelength that characterize, sorry, this kind of light is really close to the wavelength that is affecting the DNA. So for any structure, DNA structure, this UV light can penetrate till the DNA, and it causes these ruptures in this kind of bonds. So this makes the DNA to form what is called dimers, and therefore the whole process of DNA polymerase and this enzyme cannot work properly. That is the direct effect of the UV, is directing, pointing the DNA structure of any organism. So this is how basically, briefly, how the UV works. In particular, the UVC, which is is the most important light in order to produce this kind of effects in the DNA. Okay, now since we're talking and we're gonna talk about the roses, we need to understand why roses, why are they important, at least in Colombia. So the importance of floriculture. First of all, here we have a chart in which exposes the world's largest cut flower export countries. So Colombia here in green is one of the main exporters of flowers in general, right? So this means that this is a really important income to our country. And in terms of roses, Colombia is really the first exported worldwide. So it's important that Colombia exports a lot of other flowers, but the roses is basically one of the main entrance of income to our country, and in particular in the region that I work, Medellin and the surroundings are well known or well worldwide known for this kind of flowers. So yeah, we were focusing on a big entrance to our region and some problems that these kind of crops have. So what are the problems really? Well, first of all, we need engineering. We need engineering applied to agro industry. Here in Colombia, uh, unfortunately, the agro industry of this kind of flowers, roses, is really like let's say old. The engineering applied to this kind of crops is really not much, and need to have a little implementation. So we have little implementation so far of emerging technologies. Here we have in the on the right typical UV lamp, which is can be used and is already used in some developing countries. So to treat plagues, basically. So here we have the main goal is to evaluate the effect of this kind of radiation on the development of fungus. All right, that is the main concern and problem in a highly important crops in crop in this country, which is the roses. So here we have a basic method which consists only in a commercial lamp, UVC lamp, a camera to acquire just the images, uh, a segmentation program that we develop to measure the infection area, a program made in MATLAB, or it's a basic segmentation program that, that we developed. And we need a microscope with a common one, a wide field microscope to just to see some like the germination of the fungus, and the okay, the fluorescence microscopy is just to check if this kind of light can produce internally or molecular or I mean cellularly some kind of if it can produce something. It's just to check that. So this is here I'm gonna present a really easy methodology to follow this high and uh, really concerning problem in this industry. I'm, I need to say that this kind of problems is are really important to this industry because countries like United States, which is say the main importer of this kind of flowers, can put fines, economical fines, if the shipment of these roses uh, show this kind of fungus, right? So it's really important that to control this fungus and how we control it because lately uh, the importer countries that are checking also the use of chemicals. So it's really important to check if we can produce or we can have a different approach to the chemicals. So this is why this kind of approaches, these physical approaches, takes importance. So basically, this is the segmentation program that we developed in the MATLAB platform. So here the goal was to just measure. Here we have an image of a rose with the fungus. So this region that is affected, we need to measure in order to have some quantitative results. So we just developed this program, as I said, in MATLAB, and in order to just measure this area, this lesion area, and in order to compare it with the total area of the petals, for example. So here we, first of all, we developed this software, and then we just apply these doses of UVC light, and we just try different incubation times. Incubation times meaning the incubation times of the fungus on the rose, because we can control the fungus. So we put the fungus on the petals, on the roses, and we just check. We give it, we have a control, which just means that no UVC light was applied, and we have a dose here in 1080 joules per square meter, and we just check how does it work. Okay, so we check just different incubation time. This was for controlling and taking care of the time that the fungus needs to fully let's say contaminate the flower. All right, so here we can see that 24 hours of incubation time is an incubation time in which the dose applied is really taking control of this fungus. But if we check 27 hours, it's maybe too late. It's maybe too late because the fungus is already here in the day 6. You can see it a little bit, but you can see it. For flowers, it's really important that a flower is healthy. If a flower can have a little stain, it can be rejected. So it's important in this kind of product like roses, the appearance is really important. In fruits, maybe not, but in flowers, since we have to have beautiful flowers, these little stains are really problematic. So, okay, here we have a chart which is resuming the images, the data coming from the images measured with the segmentation program. So basically, the conclusion here is that 24 hours of incubation time is a time in which we can see that the UVC light works better. In 27 hours is maybe too much time for the fungus. The fungus had fully penetrate the roses, and therefore is more difficult. It's more difficult to control it. But even so, there is a significant reduction of the area of the lesion area. All right. Therefore, we choose these 2 times, 24 hours of incubation of the fungus and 27, and we apply just different doses, UVC doses. So here we have that for 24 hours, the 3 doses that we use are really controlling the fungus, and for 27 hours, really the 3 doses that we try was all right. They control a little bit the fungus, but not totally. So here we have at the bottom a chart that's resuming this, and we also try not only the fungus on the petals, but just the fungus alone. So on petri dishes, we just grow this fungus and just irradiate with UVC directly, just to check that the effect is on the fungus and not on the petals. So here we can see with these yellow arrows the germinated tubes of the fungus. This means that this kind of fungus has already they are alive, they can germinate. And here in the B chart, we have the fungus with not able to generate these germination tubes, meaning that this fungus had been really controlled. So with this chart, we just check uh, the direct effect of the UVC is just on the fungus, which is really a step that to be care, because first of all we have to just be sure that this UV do not affect the roses itself, just the fungus. So here we have this kind of experiment that really clarifies that. And this is the part of the fluorescence microscopy. This part, we have heard that maybe not only that this UV can control the fungus by meaning of affecting the DNA, but also that we heard that this UV in some previous works, that this UV light can induce the plant to produce some kind of molecules that can protect the plant for a further invasion. And one of these molecules is called chios. So this chios deposition is a molecule that acts like a shield in the membrane, the cellular membrane of the cells, of the plant cells. And these formations, these chios depositions, as I said, apparently are induced by this UVC light. Here we have some experiments that show the brighted spots are chios. So after a UVC treatment, we can see some formation of this. So not only this light just have a direct impact on the pathogen, in this case a fungi, but also it can produce this kind of molecules that are like protective for the flower, in this case, for an upcoming treat like a fungi. So this is like a second, like a secondary but a really important aspect of the treatment with this light, as a preventive, not only taking care of the fungus but also preventing for a following invasion of or any microorganism. And okay, finally we have like a prototype. We construct a prototype for using this. So we have like a UVC radiation prototype, which is a cage which we just put it in some sensors, UV sensors, movement sensors, and here we have a picture in which we have Rose just getting into the prototype. Here we have a view from the inside, and we create this kind of prototype because here we not only have like a summary of different sciences applied in here, but we also have like a formation of students. And here we had the opportunity to work with agricultural students, agricultural and engineering students, and some instrumentation engineering students too. So this was really nice because we have an interaction of multidisciplinary aspect of this project. And we can, with the instrumentation engineering students, we create this prototype. And yeah, basically we treat all, we prove this prototype and to be really useful and to be used afterwards in a large scale in the industry maybe. So as a conclusion for this kind of project, we have the effective control of this disease with a UVC, which is, again, is not invasive. It's just light, right? You need to take care a little bit of light, just wearing glasses and this, but it's by far, it's not dangerous at all compared to the chemical approaches that are traditionally used. And take advantage of the development we did and the combination of lights and images analysis for substitutes. Maybe we can use it as a substitute for traditional producers. Laboratory scale prototype was designed very low cost, just a couple of really cheap sensors, UVC sensor, and maybe movement sensor. That's it, no more than that. And this methodology shows that physical methods can be applied to agriculture and can be really reliable, environmentally friendly, and safe. And this is basically what we have with this roses project. But as I was saying before, not only the scientific contribution has made, just to taking care that take into account that interdisciplinary has been made, and we produce some papers, which is okay, is scientific results, but also the formation, as I was saying, thesis, undergraduate thesis in instrumentation and control engineering, undergraduate thesis in agricultural engineering. This is a great example of how this kind of science can be applied in a such a problem which is roses. And we have different approaches, different views of different engineers to just combine those and have finally some um, great results. So we have some ongoing projects in the same thing. So now we have just like in a scallion, on a scallion onion crops, we want to create, and we are trying this kind of prototype, but this prototype is on field. This is on field. This is not this is a pre-harvest, so we can use this kind of like pre-harvest, post-harvest. Here we have direct on crops, and we have also on bananas, just to control a hugely scary little fungus called that caused the crown rot disease in bananas worldwide. So we have here just another prototype, another project ongoing. Uh, with just, for example, here we have just the fruit. With the scallion onion, we're trying on crops. So we're trying to put this kind of approach in the middle of the process for the bananas, and we're just going with this since it's has very like good results and maybe can be applied to other kind of fungus and other microorganisms too. So that's all. I think I cover up all the mainly aspects of this project and the upcoming projects too. So I don't know if have some questions about it. And again, thanks for having me. I'm ready for any discussion or question that you may have.
Uh, Thank you, Dr. Kuhano, for that wonderful and well elaborated talk. Can you hear me?
Yes.
Okay.
Yeah, yeah.
We have a couple of questions. One is from our faculty, Ms. Harshita, that does UVC affect the shelf life, color, and sensitivity of the flower?
Yeah. I didn't show that because it's a lot of slides. But basically, yeah, we did. We check this UVC light before, so just on roses, on healthy roses, just to see if the roses were uh, at the end. I don't know, maybe cooked by this kind of light. But we make sure that we have the doses that we cannot see any of, let's say, visual effects on the color, the shell life. We try to, and we have in our paper, uh, we expose this, and we say that we try the shell life, and is some, in some cases, even prolong the shell life. So this UV is really interesting. It's not only maintaining. I mean, if you need to try before what kind of doses are not burning this these roses before checking with the fungus, but even the shell life has to prolong it a little bit. So yeah,
Yeah, thank you, Professor. I have a couple of questions. One is
yeah.
somewhere I remember reading that UV radiation at night is said to be more effective. What could be the logic behind it?
Sorry, I didn't hear it quite correctly before. There was
The
an
radiation,
interference.
UVC radiation, if it is
Yeah.
given at night time, is said to be more effective. What could be the
Okay.
logic?
Yeah, we here in this project we tried that too. But we try and we call it a dark, like a dark period of
Okay.
time. In our case, because we have heard that we read that on some previous works, there are differences, and in some works that this dark period after the UVC application, and can, like in some cases, can help this the plant, in this case roses, but we haven't heard on roses. It's just in other plants, on other fruits, that can help this UVC approach, and some others didn't show that. In our case, we tried this dark period of time, like we tried 4 hours after, and basically what we found is that in our case it did not improve. It did not improve, but it improves, it's mainly, they say that some researchers say that the plant has the time to recover this stress, because UV light is like somehow and stress for the plant. So the plant can recover for this 4 hour for this application of a dose, can recover the plant, and the microorganism may recover too, and this can add or not to the effect of the UV. In some plants, as I say, it works. Some doesn't. In our case, it did not work. I didn't show that because a lot of slides, but in our case, it did not. It did not work. It was like the same results. So maybe we need to use more, like a dark period of time. I don't know, but yeah, this is what we found.
Okay. One more question from Dr. Smriti Prabhu, that
Yeah,
as UVC is not required for the plant growth,
yeah,
it can quickly affect the plant's DNA or damage the DNA. Hence, how can its application be justified on field?
yeah. Um, Basically, this kind of light, UV light, can go deep, right? go deep and affect the DNA chain as I show. The thing here is that there are different microorganisms. So this microorganism has different cellular membranes, right? So in our case, for example, we make sure that the rows were not affected. In our case, is let's say morphologically, like we cannot see stains uh, on the plant, right? So the effect that is the DNA is being affected, and therefore the cell die, and this can be seen as a burnt let's say plant, in this case roses. In our case we didn't have that. So but we have that on the fungus. This is why we can use this, because this is affecting the fungus in a dose that only affects the fungus. It will affect also the plant, but with a higher doses that we checked before. We check the doses that burn the the roses, but from that results we just reduce this kind of these doses, and we check if those doses can affect really the fungus, and this is what we have. So of course we first need to check on plants, on crops, first the VUC that cannot or did not induce this kind of effects of burning, and then we are ready to just show on this microorganism. But the microorganism takes this light differently, because they can produce this kind of chills, and that reacts differently. In our case, the petal, the roses, let's say cells, produce this kind of chill, which is called kaios, and protects them for this kind of radiation. Meanwhile, the fungus did not. So,
Yeah,
yeah,
thank you so much, Dr. Kuhana.
okay, no problem.
I'll hand over the mic to our student representative.
Okay.
As we come to the end of this session, it is my privilege to extend gratitude towards Dr. Hiroshi Kihano for taking time out of his busy schedule and bearing with the time difference to deliver such an enlightening lecture on the application of UVC radiation in controlling the growth of pathogens. Thank you, sir.
No problem. It's been my pleasure. Thanks for the invitation. Uh, that's it. Thank you very much.
Thank you. Thank you once again, Professor.
No problem. Anytime.
Mr. Ibrahim Kamstrup Akawi is the Vice President for Data Management Systems and Standards at Novo Nordisk, Denmark. He is a computer scientist who has been working in the IT and pharma industry for 20 years. Having started his learning journey with technical roles has given him a solid technical foundation within his field of work. For the past 7 years, he has possessed leadership positions in various global companies and has been leading teams across continents and cultures. This has provided him with wonderful opportunities to travel across the globe and work with some of the best minds in the industry. His main focus is to take few but impactful decisions which enable and improve overall business goals, mainly through simplification and digitalization of the work done, keeping in mind that everything is possible. A warm welcome to you, sir.
Thank you very much, and thanks to all of you who are attending. I hope that you can hear and see me loud and clear.
Yes, very clear, sir.
That sounds good. And thanks
Yes.
for the nice introduction. I'm actually really honored to be able to speak to, a chunk of a generation that's gonna help improve the world when I get older and maybe take part in ensuring that I get the best treatments possible through utilizing the best technologies at hand. So I also have to say I'm also pleased to be speaking to some of my Indian colleagues and partners in the industry here today, because I haven't been to India for 2 years due to COVID, and usually it's actually my second home, as I call it, or maybe even I would say my third home, because it might be that I live in Denmark now, and I've lived in Denmark for the whole of my almost whole of my life. But I was born in a little country called Lebanon, and I came to Denmark in the mid 80s as a refugee together with my parents. So I got the opportunity to come here to study, to work with some really nice people, and I enjoy that, especially since that, this has been a peaceful country since we've gotten here. But I've just booked my ticket for my first trip to India in a very long time, and I'm so excited to get back to Bangalore again soon. So a little bit of my background of what I've tried. Actually, being a young kid, I started working really early. I've never been afraid of working, and I've always took pride in taking responsibility. So I started my early days as a 12-year-old paper boy, and working across different supermarkets in the local area where I lived, and then later in a bread factory. And then I started getting into my studies, starting getting jobs that are more relevant for this business. So the mobile tech industry, on data handling and data management, programming and application development on system infrastructure. And then in 2007, I started working with clinical data management. And it really struck me that this was the way to go, because I also come from a home that where my parents wanted me to be the best I could, and from my culture, it's pretty much, I think, also like the culture in India. Sometimes you have to become a doctor or a lawyer or an engineer or things like that. And of course, my parents wanted me to become a doctor, and there was only one challenge in that. I did not want to become a doctor. So it's not that the interest was not there, but I had a bigger interest in technology. So combining this job with my interest and my hobbies, I'm actually landed in the best place, I would say. So not just I get advantage of the job that I do, but actually I'm in a place where I get to help millions and millions of people and patients around the world, because I have, a keen interest and skills within this area. So there are many ways to make your impact on the world, and hopefully one day when I grow old and can look back, I can be proud of my footprint globally in the world, having helped a lot of people, right? Without being a doctor. So that's the message I try to get through to my parents also, right? There are more ways to success than just being a doctor, but there's nothing wrong in being a doctor. Good. So today I'm a vice president in the company of Novo Nordisk, and I'm part of heading up an organization for data management that employs now over 300 people. And then you would say, well, a pharmaceutical company that has over 300 data managers, how come? Well, I wouldn't be lying if I tell you we are the center of the universe in the company, because everything we do today is based on data. And that is the asset that we possess in the company. And that's why I say, everything circulates around our little hub here. So a very important function, and that's why we need to be a few colleagues. But also maybe not too many, because things also gets complicated when you are too many working on the same product at a time. But I'll get a bit more into that in a moment. So a lot of is happening into the technology sphere. Also, when you're in a pharmaceutical company, and even though we invest a lot of money in producing the right molecules to create the right product, we also invest billions right now in having all the right technologies so that we get the best products to the market in a high quality, fast pace, available to the patients worldwide. And we do that by using technology. So now I'll get into my presentation, and I will try to my best to allow time for questions at the end. And I hope that you'll take the opportunity and advantage of, I'll try to answer as good as I can. All right, so data management, us as the front runner in value creation for Novo Nordisk and our clinical trials. And even though we are a pharmaceutical company, look at this beautiful vision we have for our data, right? It says we're a global healthcare leader powered by data analytics and digital technologies. That's everything that I do in my job. So that's beautiful. And if we look up into, data, what is that? Well, it's not just ones and zeros in a long series, right? That is the output of, when you go to the doctor, you you get blood samples taken, right? There are results produced. Where do they go? Well, if you're part of a clinical study, they go to me. And I will make sure that these data can be handled, that they are cleaned, and that they can be used as part of the analysis, and also be submitted to the authorities so that they can utilize the data for their analysis, investigate what we have done, and inspect that, and then finally give us hopefully a thumbs up so we get a good, efficient product to the market for the benefit of our patients. And now I'm speaking of patients like, it's everybody else. The truth is, you all know someone at some point who is or have been a patient. I also think you can be pretty sure that one day, me, you, everybody will become a patient. And that's why I feel, when you work in the place that I do, you have an enormous responsibility to carry out your job well. But also, you have the obligation to keep developing, right? And it's not just you personally who has to develop, but it's also ensuring that the technologies are developing in the right direction. You involve yourself in the market with suppliers of technologies. You take in the right technologies for your company to make sure that you can do the best analysis, the best handling of data, the best way of collecting data, right? So, until not many years ago, we used to collect data on paper forms, right? And that's actually not really the case anymore. We hardly use paper in collecting data for clinical trials. Now we use, devices like this. This is my iPhone, where I can have apps to collect, maybe I put in data about my daily treatments that I get, about the medications that I take, the doses that I use, and so on. And that data can be transferred to data management. And, we can use that data as part of our analysis for developing new medication. There's also a lot about behaviorism data that can tell us a lot about, how do the patients behave, so that we can tailor a product, a treatment, or a drug that can help you throughout your day through certain behavioral patterns. This is also where wearables can help us, and wearables can collect now a lot of data from us. That's not a very old technology as such, but it's really ramping up and getting utilized more and more, and the functionalities are also increasing. We're also doing now a gene sequencing, and this is where we start really to get into what we call big data, because usually when I used to work on clinical trials myself, the biggest trial I could imagine would have 1 gigabyte of data, and that is data from 10,000 patients. Now one sample from one patient will produce 5 to 6 terabytes of data. So it's a totally different type of data we're getting in, and it's a totally different size of data that we're getting into these days. And then, of course, there are many more data sources. So imaging is also something you can do. And this is where you get to use your artificial intelligence, because it takes a long time to look through many images and scans, like the example here. But you can actually apply algorithms that has pattern recognitions and so on, that can help you tell, what is the activity here on this picture? Do we see a trend? Do we see a development over time when you do multiple images and so on? And that way you can, maybe target a specific part of the brain that you want to treat, and that way help us in our work and analysis. So there could be a lot of things we could get into here, but let me tell you that as we get smarter, as we get more data, we also get ambitious, because we know we can do much more. And if you look at the trends here, it looks like that we have to be, if we look benchmark from 2020 here, where we're supposed to be around 400 people. If I look just in a couple of years ahead, we should be around 800 people. And that is because if we look at down here, the number of clinical trials that we want to conduct was around 120 to 130 trials. Now we're moving towards 250 clinical studies, right? And that's because we want to keep ensuring that we have the right treatments and products for our patients. But it's also possible because we have the data to direct us where we should be going, right? So the more data we collect, the better the analysis we can make, the better treatments we can target to our patients. And remember, as I said, one day we all gonna be patients. So if I'm sloppy at my work, at one day, that's gonna bite me in the behind, right? So I better do my job well. It could also be my family members. So Novo Nordisk is very known for diabetes treatment, and I have a father who's a diabetic. So the better job I do, the better opportunities he has to live a better life with the chronic disease that he has. So the thing is, this is why I told you in the beginning, we cannot just grow like that. I mean, our pipeline should grow, of course, because the more and better products we have, the better it is for ourselves and everybody we know. But if we start doubling here, right, and the number of trials even go higher, right? So we're going to 1,000 people, 1,500 people. That's not what we're supposed to do, right? Because the word of saying, too many chefs in the kitchen will probably burn the food, and this is what we want to avoid. So instead of just having a one-to-one proportion between workload and number of employees, technology is the answer, right? And I've tried to make a little forecast here. What is it that's putting pressure on us? And if you're seeing, we are going to conduct analysis from data from an increasing number of patients in the pink circles. And if you look into the blue ones, it shows you, back in 2011, we used to collect 110 million data points. I can actually tell you, if you go back 5 more years, so 2005, 2006, we used to collect about 5 million data points, right? 5, 6 years later, we're collecting 110 million data points. We're expecting to start collecting more than 800 million data points when we get to 24, but that's not counting, all the device data from the wearables, from, all the mass production of data from our phones and so on, and not from the genomic sequencing, because if that number has to be here, I don't think I have space for all the zeros on this slide to tell you how much data we have to work with. And no matter how many people we employ, we'll never get to the bottom of looking at these data points, right? So we need smart technologies to help us. And that's why, technology is the answer to every prayer that we have. So to give you a little bit of overview of how we work and the process flow of our work, I've brought with me a little movie here that you can lean back and watch. It takes about 2 minutes and explain a little bit about my world and what I deal with on a daily basis. So please enjoy.
Data collected in studies are crucial for Novo Nordisk business and for getting new state-of-the-art therapeutics on the market. And it all starts with the clinical study protocol and study data. For subjects, their journey starts with being enrolled into the study where the data collection starts. Subject data is collected continuously throughout the study, and the digital solutions must be set up by data management. We ensure we can capture the data with the right control measures in place, and we ensure that the data is monitored, cleaned, and in control. Our presence enables Novo Nordisk's ability to make informed decisions on our study results, and prepare for submissions for getting drugs to market. Data management has a crucial role in Novo Nordisk. We enable digital empowerment of data collection for subject and study data, ongoing data quality assurance and GCP adherence, and decision-making on data results and submissions for authorities. Data management in Novo Nordisk has initiated the journey towards modernizing ways of working, and improving the digital enablement to support it. We call this the data management transformation. And the business value delivered is, data management is off the critical path, increased quality with reduced costs, and fast turnover for study changes. The data management transformation covers many initiatives, and the common focal points are speed and cost effectiveness. We ensure that data management costs and resources swifts away from operations, and towards focusing on progress of clinical studies. Selected success stories from the data management transformation are, data management no longer a bottleneck for trial setup. We save over 50 million Danish kroner in vendors' contract while reducing delivery timelines. And supported digitalization of high-profile studies, supported the EBIT implementation, showcased the reduced setup timelines for our data capture applications, and introduced the risk-based DM approach allowing us to do more, with less resources. Our critical success factors have been, replacement of legacy systems, adoption of risk-based thinking, adoption of agile in our processes, and adoption of robotic process automations.
Good. So hopefully that gave you a little insight to how we work, and hopefully the video came through with sound and everything. So
Yeah, it was good. It was good. You could hear it very clearly.
Yeah, the audio quality and everything was fine.
That sounds good. So I'll continue here. And the video you just saw here was basically representing what we do in the clinical development process, right? So we start, in the early days with research. Someone gets an idea, oh, we want to come up with a treatment like this or that. Maybe by looking at data, they try to create a molecule that mimics that activity that we want in the body, and then start testing out that molecule in a preclinical setting. So, start creating tests on maybe different tissues, on, animal rat studies, on pigs, and so on. And when they find something that looks like it works and it's safe, and most importantly, well, we start scaling it up and preparing it for clinical testing. And this is where we start to help our colleagues, our medical colleagues, design the data collection applications so we can collect the data that we want as the output of the studies that we're conducting. And we follow it all the way through into the market. So once the product has been marketed, we more or less let it go, but we keep ourselves available if someone asks for the data. If the regulatory authorities, they come and do inspections, or they have questions about the data, if they see some kind of side effects that they suspect, is that related to this drug or not? And then we help clarify some of these questions based on the data that we possess. And that's an exciting process to deal with. So our little landscape looks like this. So we start the data collection process. We do that through a variety of applications. So we have something called EDC, which is an Oracle product called, which is, EDC is electronic data capture. So instead moving away from the paper forms, now we're getting into applications. And this is like an electronic patient journal where we collect data from the patient, from the doctor at the clinical site. It could also be that we are setting up devices to collect specific data points that we want them to. That could be creating an app for, again, my iPhone or my smartwatch. We also set up databases to be able to collect data from our central labs. So this is where, when you get to the site, a clinical site, your doctor pulls out a blood sample, and that goes to the lab. But at some point, the data, as I told you before, that goes to me. And so on and so forth. And at some point, it ends up in my database, right? So that's what I possess. That's the asset that I possess, develop, and protect for the company. This is the clinical database, right? And that's where all the data gets into. Now we are, getting ready to expand on more types of data, because as the number of studies goes up, the therapeutic areas that we're looking into are increasing also and getting more diversified. We're also expecting different capabilities in order to handle these data, both from us personally, our skills, but also from our infrastructure to be able to handle it, both from a size and complexity perspective. And then when the data is here, this is where we have people who are good at setting up databases to capture the data, but they also program different checks. And this is not just about being a good programmer, but this is about understanding the data that you're doing programming for. So if I have to program checks that validates whether this data is correct, whether this data is somehow clean, I need to understand what the study is about, what the drug does, and combining these things. So my programs are working properly. So this is where you get an opportunity to combine your technical skills with science, what makes it really exciting, at least for guys like me. And then, of course, when the data is in a valid state and it's formatted in a standardized structure, we get into the visualization part, because now we have to facilitate the data so it can be used by ourselves and our scientists, right? And here, we use technologies like the Oracle Analytics Cloud, Power BI, ClickSense, and a range of visualization tools to look at the data in a smart way. And we have to do that because we can't look at all the data. I just showed you how much data we're collecting. No matter how many we are, we will need a smart tool to work on the data. And because the data is so big, eventually when we pool all the data together, we need a data lake where we can do big, heavy analysis, and also not just on data records, but also on pictures, images, scans, all these kind of things. That's heavy duty stuff. And that's something that needs to be done in the data lake. So when you combine the data from a range of studies, historically, we've conducted, thousands of studies. We've existed for 100 years, our company. So imagine the data that we're in possession of. So I'm not saying everything goes in here, but a lot can. And that allows us to maybe find new indications, new trends that we might, go back to the molecule development and then pull through this process. And that is possible because we are the one who facilitates the data, enabling exploration, development, and collaboration across our great company. And this is not a static picture. What is happening is the technologies are developing faster than ever before. So we need, for example, bright minds like yourself, young, committed, enthusiastic people who comes in, who have knowledge about the newest technologies and what we can use them for, right? To the oldies but goldies like myself, I have to unlearn and learn all the time, right? So you need to take that advantage when you get into the market as a valuable resource. You are very cherished because you don't have to unlearn a lot of bad habits first. You get directly into the newest ways of doing things. So that's a nice advantage. Again, as you would think, the overview I showed you before looks pretty smooth. It's not. It's actually really, really complicated, and there are a lot of things to it because you have all the scientific discussions on top of the technology you're supposed to master. And in order to make our life easier, we've tried to look at what are all the manual processes that we have within our work, and then we developed robots for them. So it can be done through, UiPath, Python scripts, .NET programming, and so on, and SAS programming also. So depending on what challenge you are trying to solve, you apply the right technology for it. And then we like to give them, you can see our people try to be creative once in a while. So we give them names like superheroes because, they help us so much in our daily life that they are our little superheroes, at least. And then the same thing, looking at data. Again, we can't look at all data, so we use graphics like this to give us indicators. Where do we see some trends? Where do we see some deviations? Where do we see some positive signals or negative signals in our data? And then, collaborate with our scientific colleagues about what kind of issues or opportunities do we see here, and dig a bit more into that. And that's really exciting. This is where you get to be a little bit of an investigator, play Sherlock Holmes, along with your techie brain gets stimulated. So this has not always been possible, I'll have to say then, to work in this smart way, but because you saw the trend, with the number of patients that we are going to work with to develop new medications, to the number of data points going up and so on. We made a little strategy 3 years back, and that was to help us get through all these challenging phases that we're seeing, develop a new infrastructure, and deal with all the new data that we are seeing coming towards us. So we did a little data management transformation project, and that's basically enabling us to replace our legacy systems with some new modern systems that could cope with more of the data we are expecting, but also allow for more advanced programming on top of the data, so we can really inspect and validate the data in a different way. And again, the process I showed you before, all this development process, that takes years. And we said, okay, well, at least looking at data management, we want to do a project where we can enable ourselves to get off the critical path. So we've actually done that, and it's amazing how much happens when you just start thinking and dreaming and combining those two things. So we actually really, I think in this project, and the technologies in scope, we actually made ourselves 80% more efficient than we were before. And at the same time, we saved a lot of money in the process, right? Because the more people you occupy for a longer time, our people cost, if I take an average of, salaries between India and Denmark and other people we work with around the globe, it's probably around $100,000 a year, right? And, when we are a lot of people working a long time on a specific study, that study price has an impact on how much the drug will cost for the patient. And remember, you'll be the patient one day, maybe. I'll be the patient one day, maybe, right? So we better make something that's sustainable. The same thing is, we started looking into how we could use our data lake. So NEEDLE is, our fun little abbreviation for Novo Nordisk Enterprise Data Lake. But I guess NEEDLE is fun because you work in a pharmaceutical company. But anyways, this is what had to enable us to work with big data, because we didn't have, a regular database cannot cope with these large data objects, and that we are enabled to do now. The same thing around data visualizations. We wanted to make, first of all, have a tool to help us facilitate the data, but also, make sure that the data is, the value from the data is extracted, because it's not just about collecting a lot of data. You also want to look at it in a smart way, so you turn data into information and assets to the company. And that's something we've worked heavily on. And then maybe one of the most important ones, you might think it's one of the softer, but it's actually also the keystone about being in this mindset of a learning and innovation culture. That's something that ensures that you can remain one step ahead of yourself. And why do you need that? Well, because you want to again make sure that you use the right technologies, the right processes and methods to collect data, to process data, to clean data, and basically facilitate data to the scientific part of the organization, which you are part of yourself here. So for that, also we have explored design thinking methods and played with artificial intelligence, machine learning algorithms, and robotics. So now I can say we haven't only played, because this is something that was defined some years ago. Now we actually have a small army of robots that helps us every day, and we've just started implementing the first machine learning algorithms to also help us in our daily work, and that's really something we're seeing big benefits from. So we've also started looking into how do we work in a smart way. So our colleagues in IT, they started using this SAFe methodology, which SAFe means Scaled Agile Framework for Enterprises. And this is where you work in agile release trains, and this is why you see ART here, ART. So that's the development methodology that you use usually in IT, but actually it can be used in a development setup context. So we started to work like that. And as you can see, even we're a pharma company, this is about developing systems that you see here: data analytics, data facilitation, new, and this is statistical computing environment, a system team, data lake. So a lot of what we do in a pharma company is actually about systems, technology, and data. So being aware of everything we do internally, there's also a lot that impacts us externally, because I've mentioned the regulators a few times now. But they also have expectations to us. It's not only us that are ambitious on our own behalf, but they are also. And because they also, like we do, are pro the interest of the patients, you, me, our families. And they're expecting now we start to use more data to develop better medications. So this is where the real world data comes in. And real world data can be, public registries. It can be, university studies and things like that. So all of that is something that we can acquire and combine with our regulated clinical trial data that we usually work with. And that might save us time, because if we can get some learnings from the real world data that can be acquired by us, or at least we can get access to it somehow, we don't have to conduct a clinical study to get the same data ourselves, and that also give us a head start when trying to develop new medications. The patients are also starting to be more awake and aware that they might have an influence on their health. So they want to follow their treatments, get more information about it. And this is where these devices are wonderful. And now I've pulled it up a lot of times, but it's also a very powerful device, right? If you put the right technology into it. And we started to do that much more now. There's also the healthcare value chain, again, who can be consisting of these physicians, providers of medications, and the payers. Sometimes it's yourself, sometimes it's your insurance companies or public institutions. And they also started to look at the data in ways that we do, and either gives inputs or gives challenge us on the way we work, or how we could be working, or what we should be looking into. So that's also exciting for us to have that partnership and collaboration. And then, as I've already said, the pharma industry, but also the vendors, technology vendors, have started investing. They've always invested in this industry, but now it's like never before. Not just because the industry is growing, but we've started to recognize that technology is the key answer if we want to make good medications fast. So that's wonderful. And then finally, tech companies entering life sciences in the value chain. Well, just as I said before, that has helped us enable working with big data. We couldn't do that always before, even though we wanted. But now everything is possible because we adopted the technologies. Good. So the must-win battles that we have now, considering that we claim, as I showed you in the beginning of the presentation, that we're a global healthcare leader powered by data analytics and digital technologies. And hopefully you've seen that we are now. And that will, allow us to win these battles that we set up for ourselves by, making faster, smarter products, by using automation, real-time data and analytics. And hopefully you've seen that a little bit in the slides that I shared with you. We also want increasing insights and value of the assets. And now the assets here is also the data, right? So by modeling, simulating, informed research and development, plus analytics across multiple data sources. So again, data plays and technology plays a big role. And then we want to enable precision health through omics, right? So gene sequencing, sampling, and so on, and imaging, digital biomarkers, and data analysis. And this is something that's possible now, because now we can work with terabytes and petabytes even of data. And that gives us new opportunities. But again, we need to apply our scientific thinking on those data. But the data can also help us apply, the scientific thinking. So all in all, we are well on the way, I think, for our must-win battles with the new infrastructure that we've built for ourselves, as I showed you previously. So that's basically what I chose to bring with me today. And hopefully you see some of the key takeaway messages is that we really want to drive value for the patients, thereby also the society and our company through being data science. And now I'm hoping a little bit that you want to bring some questions to the table, something you might be curious about, something you might be in doubt about, or just dying to know. And maybe I might even be able to answer. So I hope I'll start to see some questions popping in the chat. Otherwise, I could go on and speak forever, but I'd rather hear a little bit from you now.
Yes, sir. No, thank you, Mr. Ibrahim. It was really a very enriching talk. We have with us Dr. Sham as a moderator who would put up some of the questions which is being asked by the participants as well as some of the students. Over to Dr. Sham.
Thank you.
Thank you, Mr. Ibrahim. Before I think I will put the questions, I would like to echo some of the responses in the chat that it was one of the excellent presentation. That was the comment by most of the participants. And coming on to the questions, first question is, in the broader sense, the rarer diseases are generally neglected. And globally, how well is the data managed for rare diseases?
Yes, that's an excellent question. And it's true, back in the days, rare diseases was neglected significantly. And I want to say it's not just because we don't know about them or we don't think we're able to treat them, but first of all, the clinical development process takes 10 to 15 years to develop a drug, usually, sometimes even more. And back in the days, we didn't have technology like we do today, right? So some of the rare diseases have started to be treated, and we start to have some treatments develop, but not with the same pace as many of the other, you can say, generic diseases that we see. But we've actually started a pipeline called serious chronic diseases in our company. And I think what has enabled us to do that now is that we are able, for example, to do gene sequencing. And by that, we can look into, the parts of the data that comes out of it to see where is something wrong. And because we have data scientists that can do, programming on top of, applying science to their work, and working with, doctors and researchers along the way, things can happen much faster and much easier, and that makes it less complicated to produce treatments for rare diseases. So if you ask me, it's not because the willingness was not there, but it was close to impossible with the technologies at hand before, right? I mean, and I told you, before we used to look at, paper forms and data because we didn't have that much. But if you want to treat rare diseases sometimes, and you don't have many patients to test on, it makes the development process, very lengthy and very complicated, and that's not required to the same extent today. And that's why we now, whenever we could, we acquired the technologies available to enable us to start making treatments for rare diseases. So I'm pretty sure you'll get much more of that moving forward.
If I may add something, Abraham,
Mhm.
current world that we are living in, the COVID, for example, it has really changed the way the clinical industry really works, right? So that in itself is a classic example of how we are actually adapting to the current world.
Absolutely. It also showed us that, when we want to, we can actually develop treatments really fast. Now, I would say vaccines is something that has a history of, being able to be developed fast. But vaccines also do not usually go through the same, process with the authorities that we do with the regular treatments. So we have a bit more of a lengthy process and inspection process from the authorities. But when you have, a pandemic, the authorities cannot say, well, it takes the time it takes. No, they have a, global responsibility for the entire planet. So they need to push everything aside and then work only on getting, the COVID vaccines in place. All right.
We have a next question, which is the clinical data collection is the most challenging task for the research. What is your thoughts on this?
Yes, I can only echo that. Um, so you can say there are many ways, and now I've been, in the clinical development process for many years. I think, first of all, if I have to look at it from my angle, if you can motivate the researchers or the investigators at the clinical sites through, a very smooth process, right? So facilitating, an easy job for them by using the right technologies. Maybe they would prioritize the research work that you're doing. Um, if if you have applications where they can go in and, follow their work, track their data, maybe data of others who are doing similar work in the same field, combining those things, maybe that also motivates them to look more into that and, collect more data for the bigger purpose. So there are many ways to keep them motivated out there. Because we also have this challenge, but we also, we're such a big company that we have departments only looking into how can we motivate doctors to work with us on this. And sometimes they call me because they say, well, Ibrahim, can you pull up a solution where doctors can track the health of the patients that they're treating? So we came up with an application called the Health Tracker, and that's an application that the doctors are, could be using on a daily basis, and use it to facilitate the discussion with their patients, where they can sit and look at the graphs together. I mean, see, this is when you started your treatment. This is when you started to feel better, and so on, right? So they can track that visually, and I think that that's usually a big motivation to be able to work and track. And that's why, you also have these, movement trackers or step counters, right? that sometimes motivates you to walk more steps every day to stay healthy. I think that data motivates also researchers to be more engaged in the work that you collaborate with them on.
We have another question from our student. Could you give an example of being managed in the optimized DM system, which you explained in your talk?
Well, the voice was breaking up, Adele. Can you please repeat the question?
Could you give an example of big data being managed in the optimized DM system, which you explained in your talk?
Yes. Okay. So one time, this is what happened a few years ago. Someone had written a clinical study protocol where different assessments were listed, and somewhere hidden in the not hidden in the protocol, but somewhere written in the protocol, it said that we were supposed to do sampling, blood sampling from patients that had to go to lab for gene sequencing. So at that time, I only had a system that was, more or less 20 years old, and knowing that that gene sequencing data would produce again terabytes of data, I knew I couldn't handle it, and it's not just that I couldn't handle it, also I couldn't understand the data coming out, right? I mean, maybe some of you who are in the research sphere, you maybe have been trained to read records coming out of gene sequencing, but to me, I'm used to reading body weight, height of patients, vital signs, pulse, blood pressure, these kind of things, but the gene sequencing is something totally different, and it comes in a totally different volume. So I had to kind of shortcut the process for those data, and we made a space for them available in Needle, and Needle was our Novo Nordisk Enterprise Data Lake, right? So the big data, being able to handle big data, is now possible because we now do have a data lake, and we didn't have that 3 years ago, but we got it 2 years ago. So now that's been possible to work with big data, and putting the data into the lake enables us to run advanced programs on top of that data and start getting knowledge that we never that was never possible for us before, because we only had the small amounts of data collected from pulse or ECGs, doses and medications that patients were taking and so on. So now we have a totally different opportunity to work with data and do different type of analysis that wasn't possible before. So data lakes is a big gift to the industry.
There was one kind of personal question. What is your inspiration to take up this specific area of research?
Well, I think again, to me, it could have been many different areas in research, but I think Novo Nordisk is, at least when I remember this sentence from my first job interview at Novo Nordisk, I was told that Novo Nordisk is world famous in Denmark, right? And that is somehow true, because it's actually the biggest company we have in the country, and it's also the most prestigious. But now that was not it for me. For me, it was about knowing that this was the number one place I could be if I wanted to stay in Denmark and work with the capabilities that I have and the interest I have. This was the number one company that I could work with, because utilizing my skills in this company, I could help the most people ever possible. from Denmark and outwards. So you can say, why this type of research in particular? Well, I don't think we are not bound to any particular research or science, because we do treatment within diabetes, hemophilia, growth hormone, other hormonal treatments, and now we're getting into serious chronic diseases. So I mean, this is just about having the biggest opportunities ever and making the biggest footprint that I can by being at the right company at the right time, using what I do best.
There's another question. Which is the most widely used machine or coding language for
Okay.
this study, this kind
Yeah.
of study?
So if you want to take the traditional type of programming that we do here, is probably SAS programming. And I would like to see us expand much more on programming languages, because that's where I came from myself in the early days when I worked with mobile tech and databases and so on. But we have started because I also mentioned to you Python is also really winning in, and then R programming. So the R Shiny applications are also getting through a lot. So I would say probably the biggest ones in our industry right now, it's SAS, it's Python, it's R.
Okay. Last question, probably. In order to manage this gigantic amount of data, what kind of infrastructure is required? One. Second part is how long will the data be stored?
Yeah. Well, again, the infrastructure is built based on the purpose. So that's a little bit difficult to answer, but you can say some of we usually take in the data that's required to do our analysis. And sometimes the infrastructure for the data is created based on the analysis that you want to do. So you have a pool of data lying around somewhere that you then take into the company and store into the lake. But then you might take pieces of that data and put into a specific infrastructure depending on your needs, right? And then there was a second question to it.
How long will the data be stored?
Yeah.
Whatever data
So
collected.
yeah, that's a good question. So we are obliged to keep the data for many years, and we actually have to keep the data for at least 15 years after we take the product off the market. So probably our data will be lying around for 50, 60, 70 years. That's a pretty long time.
I think we are done with the questions. Thank you, Mr. Ibrahim, for walking us through wonderfully in the world of data management and analysis. Now, I would like to hand over the mic to our student representative, Ms. Athira. Over to you.
Yes, Mr. Ibrahim, it was a great talk. Mr. Vijendra also. It was really enriching, and I hope all my students have got to take away a lot of keynote values of data management, and hope we look forward to work together. And meanwhile, I would just pass on to my final year student for vote of thanks for such an enriching talk.
Thank you, and thanks for giving me this opportunity. I hope it gave you a little bit of inspiration for your further work, and to complete your studies and see that there's a wealth of opportunities for you when you're done. And I hope some of us get the opportunity to work together in the future. Because one thing I forgot to tell you is that 70% of my colleagues, they actually sit in India. So there's a good opportunity for all of you here. So thank you and enjoy the rest of the day.
Yeah, thank you. Thank you very much, Mr. Ibrahim.
It is my privilege to extend gratitude to Mr. Ibrahim for taking time out of his busy schedule and bearing with the time difference to deliver such an enlightening lecture on the recent innovative trends involving data science and analytical tools in clinical data management. On a personal note, as a biotechnology student studying clinical research and data management as a subject, this lecture has given us a deep insight on how data management works on an industrial scale and has widened our understanding on the underlying concepts in this field. Thank you very much, sir.
Yes, great, Vijendra. Thank you a lot.
Thank you. Thank you so much. And just before we drop out, if I may say something, I still remember the year 2010 when one of our senior who had come down to our college just to speak about data management, but it was him who actually exposed me to this world. And then I thought it would be really nice if someone big like Ibrahim can really influence a generation, as he said in the beginning. And thank you so much, Ibrahim, for accepting this and doing this wonderfully and in the simplest of the languages.
Thank you, sir, and have a good day. Thank you,
Yeah,
Vijendra. Have a good day.
you too, guys. Bye.
Bye. Happy with your questions.
Session. She may be joining us a bit later, and I'll be with you during the proceedings. We are in for the third lecture, keynote lecture for the series, which is presented by Ms. Alina Samkina. She is a Deputy Director, Chief Commercial Officer at VG Koplin Radium Institute of Rosatom Corporation, St. Petersburg, Russia. And she will be talking on the biotechnologies in radiopharmaceuticals development by example of recombinant alpha fetoprotein as a vector in targeted radionuclide therapy and diagnostics. She has immense amount of experience in the field of radiopharmaceuticals and targeted therapy as well as diagnostics. With this brief introduction, I request Ms. Alina Samkina to take over the stage. Over to you, ma'am.
Hello, can you hear me well?
Hello. Yes, we are able to hear you.
Okay, good. I'm sorry I was a little bit late. Can you share my presentation which I just sent to the email which I received from Dr. Ujwal? Sorry if I pronounce his name in incorrect
No,
form.
yeah, it's right.
Or shall I
We
do
will
it myself?
sure we'll do that. Just give us a couple of minutes, ma'am. ಉಜ್ವಲಿಗೆ ಕಳಿಸಿದಾರಂತೆ PPT.
Also, maybe I can try to do it myself if
I think that
that
would be
is.
better. While we search, I think that would be a better option also.
Could you please guide me? Shall I just put the bottom to start
There
showing,
is a,
right?
there is an up arrow button in the. I mean, once you open the PowerPoint, there is a. Okay, we are accessing the presentation, ma'am. Just a minute. We will present it for you. We are in the process of downloading and presenting it. If you can give us a minute.
Actually, I'm not a specialist in biotechnology or biochemistry, so please accept my apologize if I make any mistakes. Please feel free to correct me. I would be just very glad. Our institute is specialized in different spheres connected to radioactivity and its application for different purposes. This is Radium Institute
I think
named.
the presentation is now being shared, ma'am.
Yes, yes,
Are you
yes.
able
I
to
can
see
see
that?
it.
Yeah. Yes, yes. Thank you, ma'am.
Thank you very much.
Yeah.
So our institute, Radium Institute, named after a great scientist, Mr. Chlopin, was founded by academician Vladimir Vernadsky. He is a very famous scientist, well known all over the world, and he expected radioactivity to have a lot of different application and to serve for common good in future in absolutely different technologies, and especially with the power that is hidden inside of the nuclear of the atom. So Vladimir Vernadsky founded our institute 99 years ago. So next year, that will be 100 years of our institute. And in our institute, there are several departments or divisions which are dealing with absolutely different applications of radioactivity. So in our institute, we are making research of fuel cycle and fission technologies. We are making research in isotopes enrichment, including radioisotopes enrichment. We are running different R&D and investigational work in radiochemistry, geochemistry, radioecology, and also there are several production departments and department of radiopharmaceuticals development and production. And recently we started absolutely new work for us, which is connected to the sphere of biotechnologies. So I will make a short description of the approach. I'm not sure if all of you work with radiopharmaceuticals, and if all of you know which tool can be founded for biotechnology using different radioisotopes. And the first slide, how can I go to the second slide, or can you help me with this? Yeah, thank you. So I will make a general description of this tool, which I spoke about. This tool that help physicians and scientists to understand human body much better, and to see how our tissues, how our organs functioning. What I mean, there are two main applications of radioactivity for medicine. Of course, there are a lot of other applications. I will not talk about them today. I will speak only about one particular question. So, two main application which I'm going to discuss today. There are diagnostics and therapy. And when we speak about diagnostic, radioactivity provide us unique solution. What means unique? It means that we do not see a morphological picture of our tissues. We can see how it works, if it is alive or not. What is the metabolism? Does it have any problems, any disorders, or maybe specific behavior? This can be shown only with help of radiodiagnostics. Another application is radiotherapy. That is actually not that radiotherapy, external radiotherapy, which we can find in many hospitals. When we speak about external source of radioactivity, it can be gamma irradiation, it can be proton irradiation, and which is used for cancer treatment, for example. We speak about radiotherapy, which is target radiotherapy, or sometimes it calls nanosurgery. So we speak about molecular therapy, molecular diagnostics and molecular therapy. So how can we construct this tool to see on the molecular level the function of our organism and our tissues? How can we treat it on this level? We should construct a molecule which can specifically come to particular tissue, to particular cells, and radioisotope which is connected to this molecule can show us, if this is diagnostic, it can show us the localization of this tissue, or even a very small amount of cells which cannot be located with other methods. So, for example, very small metastases can be detected only with radiopharmaceuticals, only with this precise method of molecular imaging. So when we construct this molecule, which can specifically target the cell, or when we try to destroy the cell, this particular cell, we should take into account 2 points. We should choose the best radionuclide, and we should choose the best vector for these purposes. I'm not going to describe all this range of radionuclides which are used and all their characteristics which help us to understand which radionuclide can be used for which purposes. I just tell in general that there are a wide range of radionuclides, and all these years, maybe roughly 80 years of application radioactivity for medicine, there was type of change from one radionuclides to other radionuclides, and it was type of natural selection of radionuclides which are best for nuclear medicine. And if any of you are interested in this information, I can provide you more details. Please feel free to write me, and that's not a problem. Because I personally think that this is perfect tool, and this is perfect tool for biotechnology, definitely. So if we speak about radiotherapy, this is vectorized radiotherapy. It means that our molecule should specifically target that cells which we are interested in, should specifically locate there, and should be accumulates there. Also, it should be stable. And we should be sure that our radioisotope will be delivered to the cell, for example, cancer cell. We should be sure that this molecule doesn't destroy our organism as well. So there are several approaches how we choose these molecules. There are metabolic radiotherapy. For example, we can use iodine, and this is one of the most famous radioisotope which is used for radiotherapy. Of course, this is because of its metabolic characteristics and accumulation in thyroid tissues. So also, there are other methods like radioimmunotherapy, when, for example, antibody or maybe fragment of antibody is used as a vector. And this vector delivers our radioisotope to the specific cancer cells with specific receptors. And this is a very popular mechanism, which is now under investigation in many countries, and clinical trials are running in many countries right now. Also, there is peptide receptor therapy, which is also very popular, and I will speak today a little bit about it. So, mostly as vectors, there are used antibodies and fragments, peptides, aptamers, scaffolds proteins, and small molecules. Different molecules. Actually, there are a wide range of molecules which people try to use as vector to deliver radioisotope. There are a lot of complications of these technologies. I noticed in this slide this complications, but that benefits for patients, of course, are the most important. So, per my understanding, per my view, what is the most important benefit for patients? The first, that is, of course, efficiency. This is extremely efficient compared to chemotherapy or even other methods. Extremely efficient method. Very few side effects, and not strong. There is other very important benefit. This radioisotope, which is used for radiotherapy, it's type of toxic agent for these cells to destroy them, and this type of toxicity is invisible for cancer. What does it mean? If we speak about drug resistance, if we speak about genetic reprogramming of cancer cells, when we use conventional or even target chemotherapy, anyway, our cancer cells definitely would need more and more strong and more toxic drug with each next line of therapy. That is not necessarily for radiotherapy. There are a lot of questions which do not still have answers, but probably that is because of microenvironmental changing, maybe because of some other effects. But cancer cells sometimes disappeared completely after several treatments, and there is no problem like more and more aggressive cancer after each treatment. Okay, I will show you the pictures about this situation, which show these results. Radionuclides, which are used and shown on this slide, can be absolutely different, and there are a lot of methods to produce them. And for example, in our institute, we use all of these methods, and we have a wide range of different radioisotopes for different applications, not only for radiopharmaceuticals or for biochemistry. Mostly, we use them for other industrial applications. There are approaches when you can use same chemical element, for diagnostics and for therapy. For example, like I told you about iodine, or there are other elements which can be used in the same way. I mean that if we can use iodine 131 for thyroid treatment, we can use iodine 123 for diagnostics. And we can see the result of our treatment, or we can see it and watch it with very good understanding of what is going on inside of the organism. If we label our biological molecule, with other radioisotope, for example, scandium or terbium or other radioisotopes, we can also construct two molecules for diagnostics and for therapy, which can work in the same way and target our tissue or some cells which we are investigating, and then treat these cancer cells if we speak about cancer. Can you please move to the next slide? So that is the result. One of the methods, peptide receptor radionuclide therapy. Now this drug is that was acquired. This company was acquired by Novartis Corporation, and now this drug received marketing authorization, as you see, in Europe 4 years ago, in the USA 3 years ago. You can see the result. If we speak about some conventional standard approach, that is absolutely completely different result, completely different efficiency, completely different side effects and everything. I didn't include here a photo of patients, but these photos which shown by the doctor who was a pioneer of this peptide receptor radionuclide therapy impress a lot. So very complicated patients. You should understand that this is usually that is metastatic cancers and very bad patients, and this complete response, which happens rather often, impress a lot. So there is nothing which can be compared right now. So that is the first drug, the first radiopharmaceutical of this type, which has already received approval in many countries. Can you please show the next slide? That is another example. This is PSMA targeted radioligand therapy. So it's used for advanced metastatic prostate cancer. And you can see these photos of the patient. That is photos made with help of nuclear medicine technologies. So you can see the first photo, and you can see metastasis on these photos. On the first photo, there are PSA on the level 4.6. The second photo, you can see same patient 1 year later, and such examples, and this therapy is used all over the world. Probably all of you know about this. So I'm sorry if that is something to repeat for you, but I can say that in many countries, many doctors, many biotechnologists do not know about these methods at all. Could you please show the next slide? The most problem is that for the examples which I noticed before, there were actually two applications neuroendocrine tumor and prostate cancer. But there are a lot of different type of cancers, of course, and there is a dream to find some universal targets and, of course, universal vectors, which could help to target it. So on this slide, you can see the photo which was called the image of the year during SNMMI conference. It's international conference in the USA. And you can see this result when was used the fibroblast activation protein inhibitor, FAPI. And this radio tracer, single radio tracer, can be used to diagnostics a lot of types of cancer. In their publication, they say it's nearly 30 types of cancer. Of course, there is a very big interest if this vector can be used for treatment, for therapy, or not. This question means that there should be done a big research of stability, of biodistribution in vivo. And of course, when we speak about therapy application, it's more complicated and it's more expensive compared to diagnostics applications. And finally, what we are doing now in our institute. Can you please show the next slide? This is the work which is done together with several biotechnological companies. This is a investigation of alpha-fetoprotein, a well-known protein. Investigation of this protein as a vector to target different types of cancer. So what is promising? First of all, there are a wide range of cancers with overexpression of receptors for alpha-fetoprotein. This information is well known, and this protein is well known for many years. So this is something understandable, and the only one issue which have been a problem, a headache for many years, is that there was no one high-scale production of this protein in the world. So there are several recombinant alpha-fetoprotein manufacturing sites, but the self-cost and the price of this protein is extremely high. Of course, there is a source which is natural, not recombinant, but it's also limited, expensive, and not pure. Also, there is other aspects which ethical aspects as well. So what changed in the recent time, in the past years? Right now, there are two independent methods of high-scale production of alpha-fetoprotein in Russia. And as we know, as we read in publications, there are another method which is used in China. And these people also receive very promising results investigating this protein as a vector. What is going on in Russia right now with this protein? There has been two independent, not connected researches. One is about application of alpha-fetoprotein for a wide range of diseases not connected to oncology. But if we speak about our application, since we plan to use alpha-fetoprotein for cancer treatment, we concentrate on its properties as a vector for cancer. And we received, not our institute, but the group which we are working with, received very promising results in preclinical trials, and clinical trials have also started. So the proof of concept when alpha-fetoprotein can be used as a vector for toxic agent. In that preclinical trials and clinical trials, as a toxic agent, there is the use of actinomycin. Sorry if I pronounce it not in correct way in English. Actinomycin, cytostatic composition, and absolutely in the same way, with, of course, several differences connected to radioactivity. We are now running trials to synthesize a molecule where alpha-fetoprotein is used as a vector and radionuclide is used as a toxic agent. So all the results which are received right now in experiments in vitro, which we do not consider as a very interesting results, but also in vivo, which we consider as interesting results, shows, I may say, promising applications of this vector. And right now there are several non-solved questions, non-solved problems, like complicated labeling, connected also to complicated quality control of the final molecule, stability studies. I mean stability studies when you should be sure that your vector is able to deliver a toxic agent as a radionuclide to the particular cell. And of course, there is another question which is also complicated. That is the question of other tissues, not cancer cells, but other cells, which also have receptors for alpha-fetoprotein. So that is the problems that we are investigating and solving in our research. And we would be glad if any of your specialists is interested to participate in this research. So I tried to make it as short as possible. I'm very sorry if that was not clear for maybe some reasons. And if you have any questions, or maybe if you need, if you are interested to receive any additional information, please feel free to contact me. And there is my friend, Dr. Afsal from S&T Healthcare. So he is very good radiopharmacist who, of course, can answer some other questions, of course, connected to this problem.
Thank you, Ms. Alina, for that wonderful talk. We have a couple of questions for you, if I could put it up. First one is, could the radioisotope itself have an effect on the molecule after being labeled?
Yes, of course.
Yeah.
Yes. That is one of the main problem. If you use such radioisotope like iodine, you can easily connect this radioisotope to mostly, maybe to all biomolecules. And if we speak about other radioisotopes like metal radioisotopes or other with another chemistry, you would need to use probably chelating agent. you would need to use linker, and of course, this construction makes influence on the biomolecule, and this is one of the most important and most complicated question. So you should be sure that your diagnostic molecule modified with radioisotope has same biodistribution as therapeutic molecule.
Okay. Then we have the second question. What about the nephrotoxicity of radioisotopes which are being used in the diagnostics or therapy?
Both these questions were notified in my presentation. I didn't stop on this question. If you go to the, probably that is the first slide.
Okay.
Yes, that is the first slide. So you can see here, there is a problem which should be taken in account concerning renal and liver uptake. So if we speak about nephrotoxicity, it means that this radioisotope, not only radioisotope, all the molecule has sorry, I don't know how to say it in English. So it has renal uptake, and this radiopharmaceutical can damage and can have nephrotoxicity. So one of the important question is to use molecules which would have not renal but liver uptake. And when we speak about liver uptake, in this case, that would be the better way, and there is no nephrotoxicity in this case. And this, of course, is taken into account for many people who are developing radiopharmaceuticals. So you are correct. This is important question, and it is always under consideration when you develop new radiopharmaceutical.
Okay, and the last diagnostics or therapy.
I'm sorry, I didn't understand the beginning of your question. What is?
What would be the criteria for choosing the best radioisotopes for the therapy or diagnostic? Yeah.
Yes, I understood. Criteria. You can see on the first slide as well. There is written type of radiation, energy, and lifetime. That is all these are criteria for radioisotopes, and chemistry. So if we speak about chemistry, that's your first question. If radioisotope modify or non-modify the molecule. When you use iodine, this is the way how to reduce modification, how to minimize this influence. But if we speak about other radionuclides, that is another chemistry. So that is the first criteria. If you can use, if this chemistry change your biodistribution, your specificity, or does not change. Another criteria is type of radiation. If you want to make diagnostic molecule, you should use gamma emitters, or you should use positron emitters. If you use gamma emitters, that is SPECT technology. If you use positron emitters, that is PET technology. Of course, very important is lifetime. It should be the best for your case. It should be adapted for biodistribution of the molecule. For example, if you have big molecule like antibody, you should take radionuclide which half-life roughly 2 days or 3 days or even more. If you take small molecule, you should take radionuclide which live much shortly. Yes, so it has a shorter lifetime, but also very soft radiation because of this renal uptake and nephrotoxicity. So if this is very soft, there is no problem for renal at all. And if you need radioisotope for treatment application, of course you should use electron emitters, beta emitters, or alpha emitters, which are even better. And for therapeutic application, you should also understand all these complicated things like lifetime, the energy, and possible renal uptake. So that is all these criteria are taken in account to understand what is the best way.
Okay. Thank you so much, Ms. Alina. I request Dr. Ujjwal to take over the session now. Over to you, Dr. Ujjwal.
Yeah, well, good evening, Miss Alina.
Good evening.
I'm delighted to have you. And we had a good interaction. Your talk was really enriching. Dr. Afzal, would you like to add something? We have industrial experts.
Thanks, Ujjwal. We don't have, this is a very good presentation. Thanks, Alina, for sharing your inputs to Nitte-Bio group. Definitely, nuclear medicine connected to biotechnology is a growing field. And it is something like we are expecting the researchers to investigate more in this aspect. There are many questions. I heard all the questions, and Alina answered the question. So we would like to share more knowledge regarding nuclear medicine with the Nitte students and Nitte team anytime. You all can approach us. Our contact details are there with Dr. Ujjwal. So we are happy to help you all. Thank you.
Thank you, Alina, for all your support and your enriched guidelines and your talk. I am very delighted to have you for this conference. Thank you once again from the team of Biotechnology Engineering and MIT Nitte.
Thank you very much. Thank you.
Thank you. Thank you. Have a good day.
Thank you all.
Have a good day.